Hello and welcome to our poster. My name is Rune Berg. In this poster we will discuss two parts. One is the discovery of the population activity behind rhythmic motor activity in the spinal cord is executing a rotational dynamics. The second part is development of a new theory that can explain rotational dynamics and a number of other unresolved issues in motor control. So the open questions are generally how is the rhythm being generated? How can it be modulated, both the speed and the force independently? And how can multiple motor patterns be generated by the same network? The experiments involved in understanding spinal motor circuits have rarely involved the neuronal population activity because it's rather difficult to access the neurons in the spinal cord because of its flexibility. But we use the turtle spinal cord because it has a mechanical advantage among other things. And here you can see a decapitated turtle performing a scratch reflex that's induced by a sensory input. It can produce multiple different distinct motor patterns, for example, pocket scratching and rostral scratching, as shown here. We use multi-electrode arrays from Neuroscience. We insert 256 channels into the lumbar spinal cord of the turtle while it's performing this motor activity. This is what it looks like, the experiments. And here's a post hoc immunohistochemistry where you can see the location of the electrodes. You can also see all the motor neurons stained with the chat, and then there's interneurons with new N. Here's a typical recording. You can see at the top we record the motor nerve output where it's performing one trial of a scratch reflex, 5 cycles in this case. And below you can see about 256 neurons shown in a raster. And below you can see the estimated firing rate of these neurons. And it looks rather incomprehensible. But there's some rhythmicity to it. And if we sort these neurons according to the phase with respect to one of the nerves, it becomes much more clear what is going on. As you can see, it seems like there's a sequential activity where the population as a whole is cycling through all phases. There's a name for this type of activity, and it's called rotation. And as you can see here, you have the sound is the population spiking. We also performed a PCA to better describe this rotational dynamics. And it had a very low dimension, more or less 2-dimensional, where it's cycling, it's rotating around in this plane. Here's shown multiple trials performing scratching, and it's repeating this rotational dynamic. On the left you can see the sequential activity. It's always the same sorting that we show. In the middle you can see the distribution of phases across the population. There's no particular phase that's preferred, and all phases are represented. On the left, you can see the population rotational dynamics. And what you notice is that it's changing its radius both within a single trial, but also across trials. So we looked more carefully into this, and we noticed that when we have a larger radius of rotation, that the nerve output is also larger. So there's this nice correlation between the radius of the rotation and then the intended force by the network. Why is rotation important? If we take a look at the traditional view of spinal motor circuits, it's considered that there's these modules that are alternating in activity. So you have one module or group of neurons that are active at one time, and then it's flip-flopping over to another. It's called alternation, and that can explain this alternating activity you see in the nerves. But what it seems that we're observing in the population activity is rather this sequential or rotation in the network. And you can explain alternating muscle output from a rotating population dynamics, but you can't explain rotational dynamics by a alternating modular network. So there's a need for a new theory, and we have conceived a new theory which we call the balanced sequence generator. It's basically based on a sparse random networks that are consist of recurrent, excitatory, and inhibitory neurons in a balanced network. And we implemented in models where we use rate-based activity. And the network is activated by simple modulatory input from, say, a supraspinal center, or it can also be sensory input. So here's shown an experiment on the left, similar to what I showed earlier, and then a model implementation shown on the right. And what we notice from such a simple network is that it's executing rotational dynamics, very similar to what we see in experiments. We also notice that depending on how we activate the network, we can actually induce multiple different motor patterns, as shown here, where we are switching between the two motor patterns I was talking about earlier, which is the pocket scratching and the rostral scratching. So it's starting with the pocket scratching, and then it's changing to the rostral scratching. And the input you see is on the top left is that a very, very small subset of cells are either activated or inhibited, and that can be and changing between these activations, you can induce these different motor activity. The other thing that's interesting is that the sequential activity is more or less the same in both patterns, even though the motor output is distinctly different. So the rotational dynamics seem to be more or less conserved in the model across different behaviors, and it's also conserved in the experiments. With that, I would like to thank my colleagues, and also in particular Henrik Lindén, who's done a lot of work with the model. If you have questions or comments, please come to the interview session on Tuesday. Otherwise, enjoy your conference, and thank you for listening.